All right, let's get started again. In about 45 minutes. Lunch. I do want to tell you about read formats. Essentially, the format in which the reads come out of a sequencer or come out of the alignment after the sequencing and the alignment step. So it's basically the way the read format situation has become is there are two clear winners. There are also formats that used to be used and less used. Again, it also depends on which alignment tools is winning in terms of adoption and use. So the SAM format is what comes out of both BWA and Bowtie and many other tools now. This is an example here of the SAM format. Again, it's all in the same file. Each line corresponds to a reader. And each of the lines now tells you something about what the aligner did and where in the genome the aligner placed each of the reads. The format contains a lot of information from the original FASTQ format. This was there, the read is there, the quality scores is there. The rest is basically used by the aligner, or produced by the aligner. The key information here is the chromosomal position this is chromosome and the chromosomal position here that tells you where your read is being mapped. There are additional information that are useful when it comes to mutation detection, basically telling you about how the read is different from the reference genome. And it's captured in some format in two main pieces of information. This code here, which is called the cigar string, it basically is dividing your read in multiple blocks, and each of the block essentially corresponds to a different matching structure. So when you see a cigar string like this, and I don't know if any of those reads has this one, but when you see a cigar string like this, what it tells you again, is that you have multiple blocks. What this is telling you is that in your read, the 5 first base pairs were actually matching perfectly, right? Were actually matches. And then you had a block that corresponded to an insertion in your read, in the alignment. So in this particular case, this is an insertion of about more than 3,000 nucleotides. And then you have another block that corresponds to the other end of a read that is also matching to a different place. This is useful because it basically tells you that this is typically a situation that occurs when you have a splice junction, when you have a read that's been split into two subreads and they map reasonably far away from each other with a long gap. And this gap is essentially corresponds to an intron typically. And again, you have that information within the reads, within the format that's coming out of the read format is coming out of the sequencer. So it's nice to have, it tells you, which of the reads have been split by the aligner to align them to take into account splicing, for example. The MD tag tells you something about mismatches or inserts or short inserts or the identity of those mismatches, the identity of the inserts. Basically, you have a situation like this where you have this is a short, again, this is kind of a block structure. What this is telling you here is that in this particular read with this MD tag, you had, this should be 4, you had 4 matches here, in the 4 perfect matches between the read and the reference genome. And then you had a nucleotide here that was a T in the reader, but it's not T in the reference genome. So basically, this format here is highlighting the differences between the read and the reference. And then it tells you that you have 46 matches again. So a combination of these two structures here is basically describing how the read is different from the reference and the kind of operations that the aligner had to make to split some of those reads. So that's useful information when you look at this format here. There's also additional information which is a bit more aligner specific. So for example, BWA tells you a read is a unique mapper or maps to multiple places. It's going to add a tag towards the end of a reads with XAAU if it's a unique mapper, XDAR if it matches to multiple locations. These tags are actually aligner specific. At least some of these are aligner specific. The cigar string and the MD tag are actually reasonably common across aligners, but this is more tool specific. So what that means in practice is that depending on if you want to do statistics on some of these reads and you read them in R, in other programs, and analyze them and look for the ones that are mapped into a unique place, you would have to be careful about what program was used to align the reads, because the information about this unique mappability feature is encoded in different ways across different incarnations of the same format. It's still the same format, but it's kind of flexible in terms of some of the information that it contains, and therefore you have to be careful about knowing which aligner was used to produce those files. This is paired end SAM, where each read is now paired, and this is, let me see if we can, I mean, it's basically the same type of format, except that you have the two reads, that corresponds to the same DNA fragment. Depending on whether the format is sorted or not, I mean, these two reads are next to each other. The key here is that one read has information about the other one, and this information is contained, let me see, I think the information is contained here. So this is here, for this particular read here, this is the location of the genome of that read. This is the location of the other read that matched the sort of the paired read on the genome. So essentially, each read has information about its paired read that can help you in terms of some operations. So the BAM format is the binary equivalent of the SAM format. There are tools that allow you to convert SAM into BAM, typically SAM tools. It's nice because it's compressed, so it really shrinks the size of your SAM files by a lot. Actually, the nicest aspect of it is that it's indexable. Remember, we talked about indexing. It's the same idea. Every time you hear about indexing, it basically, when it comes to format, to files, it basically is telling you that you create a data structure that allows you to query specific locations, specific regions, specific subset of reads really, really quickly. So for example, once you have a BAM format that's indexed, you can actually very quickly access the subset of reads and map to chromosome 10, position 10,000, 20,000. That's very quick to access only those reads and look at them and do some statistics. This is what indexing allows you to do, to really quickly access fractions of your file, as opposed to having to scan the entire file line by line, until you get to the point where you have the region of interest. Okay. In fact, once you index your BAM file, you can actually upload these BAM files to the genome browser. And this is what you will see in the genome browser in terms of if you were to upload some of these BAM files. You get short reads that map the locations. Because reads can map on either orientation, forward or reverse, that information is maintained, is contained in this plot here. These are the I think forward mapping reads, and the blue ones are reverse mapping reads. Okay. This is useful because the fraction of forward and reverse reads should be about the same, and you can see here that it is indeed about the same. If you see unequal fraction, it can tell you that you have artifacts, and that you should be careful. So this is uploading the reads to the genome browser. Again, very useful as a way to visualize. You don't have to use the genome browser. IGV is a good option to visualize your reads as well. But the genome browser is nice because it has all these tracks. So what you can do is to integrate visually your reads with everything else that's known about being found, the encode, for example, multiple databases. Basically allows you to do kind of visual integration, which is very, very powerful. And not always as easy in IGV. Questions about this? Yeah.
A general question about, and this is a good example, you see a lot of aligning to the introns.
Yeah.
curious, you see so much light, you see more light in the intron than you do the exon.
Yeah, so this, like you say, it's a theme. In a seq, that you see so much, transcription in the introns, while in theory these introns, shouldn't appear in a seq. Data, there are multiple ways to explain this, and none of them is necessarily particularly good, but one of them is that not all introns are processed right away. Some introns are processed a little bit later, and somehow they find their way to the INA-seq data because they are sequenced and processed. I think that's one possibility. Another possibility is that what we know about the exons is only partially right, and then there might actually be a huge exon here that's not in the database, but may still actually may still exist and be captured by this particular experiment. That's another explanation for this. But you do see a lot of intronic, large number of intronic reads in INA-seq. It's true. I do, and every time, I give this kind of talks and you emphasize, visualization, is key. We're talking about, extremely highly complex data sets, and you should take every possible steps to visualize your data. It's absolutely essential that you try to visualize your data in as many ways as you can. You can't really, you can't look at files of reads, and infer anything useful, from these format. You can look at this and generate hypotheses, have ideas, just like discussion that's coming out of looking at this picture here in the context of this intron here. Visualization is really, really key to look at complex data sets and identifying artifacts, getting new ideas. So the genome browser is a key tool for visualization of genomic data. IGV again is another tool, but visualization is extremely important when it comes to these very complex data sets, especially. So this is SAMtools. This is a package that's very often used. Pretty much everybody uses SAMtools as a way to do operations on short reads. SAMtools allows you to convert SAM to BAM, allows you to sort BAM files, index BAM files, and convert BAM to SAM. Also allows you to visualize BAM files. BAM files are in binary formats. You can't look at them using a text editor, but you can view them using SAMtools. View actually can work on something like this. Yeah. And there is nicely an interface to SAMtools in R that Matt is going to tell you about tomorrow. And he's going to show you how to use R SAMtools on some BAM files. That's useful as a way to integrate SAMtools into your workflows. You can still do a lot of things, in R. You can call programs, command line programs in R, so you can integrate command line programs in R by using the system command, which again allows you to execute external programs in R. But the SAMtools package allows better integration. It allows integration of SAMtools using some of the other tools for interval operations, like genomic range or high range. That's very useful. There are also tools in SAMtools that allow you to get statistics on your reads. This is just one way to get statistics. It gives you the number of reads that are mapped, the number of reads that are paired, numbers that are properly paired, and so on and so forth. Again, useful to get statistics. It also allows you to do pileups, which is a let's say Pullman's version of his genome browser plots that I was showing you. Not so much Pullman, but different. Essentially, not very prone to visualization of entire regions, but it does allow you to look at specific positions, and that's actually useful when it comes to looking for mutations, looking for a certain number of nucleotides in position that don't match the reference genome and could constitute a mutation. and you also have quality scores for across many nucleotides. Some codes, I mean, for locations where you have a beginning of a read or end of a read. And again, you know, this is something that can be useful. I mean, the only problem is that it creates, you know, gigantic files that, you know, is not always easy to manage, but it's useful. There's the uppercase and lowercase nucleotides here, and these are basically nucleotides that come from reads with different orientation, reverse of all reads. So yeah, so we discussed this, but SIME-tool does allow you to collapse clonal reads. That's actually not the tool that's mostly used for this, because it's good, but it's not pair-then aware. So Picard is another tool that people use to collapse clonal reads, and it's actually pair-then aware. But even so, I mean, you know, if you have single-end reads, you can actually use this command here as a way to collapse clonal reads. Again, clonal reads are, you know, things like this, reads that map to the same location, same orientation that you want to collapse into one frequently for many applications. This is, let's talk about secondary analysis. So by which, this here will mean transcript level quantification and mutation analysis. I'll go fast on this because that's not necessarily the theme of the course, but one key concept in terms of transcript quantification is the concept of RPKM, which you can also often hear as FPKM, which is pretty much the same thing. The key problem is that you know, when you want to estimate transcript levels for a given gene from hide and seek reads, for example, you're going to take your short reads, map them to the genome, you look at a given gene, and you count how many reads map to that gene. Right? So why is it that just counting is not good? Is it good? I mean, it's not good enough. Why is it that it's not good enough? That's right. So if you have a very long transcript, you're going to get more reads just because you have more room, right? So you do have to normalize by the size of your transcript, right? So, and what else do you have to do? Now imagine a situation where, you know, you sequence a sample and you get, let's say, 100 million reads, right? Then you sequence another sample that you want to compare to the first one, and you get 200 million reads, right? If you don't correct for the fact that you got vastly different number of reads between the two runs, you know, all of your numbers in the second run are going to be double the ones in the first run, right? So you do need to also correct for the fact that you have different number of reads across experiments if you want to be able to compare experiments. And so these two normalizations basically go into this RPKM measure, which a lot of people use, which is basically the number of reads per kilobase of transcript per million reads. Okay? And again, you know, the formula is reasonably easy. You count how many reads map to a transcript. You divide by the length of the transcript. You divide by 1,000 just because you don't want these numbers to be the ratios to be too small, and that's why you have the kilobase of transcripts. And then you divide by the total number of mapped reads per sample divided by 1 million. That gives you a number which is an RPKM number, which you can compare between experiments because it's normalized, at least when it comes to total number of reads. And then that a lot of people use as a way to quantify expression levels. So this is RPKM, and this is something that is useful. Yeah?
How do you deal with splice and non-splice transcript? Which one do you use?
Yes, they basically would have different lengths, that's right. So you would have, this is a bit more complicated. This actually, I was kind of getting to this. The RPKM is actually sort of more easily applicable to genes when you have, you know, a single unit, right? And you can compare between experiments. So if you want to do a splicing aware analysis, What you would want to do is actually use a software that's a bit more sophisticated than just simply counting as we did. And some of the software do essentially, and Cufflinks is one of those, is to do a probabilistic assignment of reads. It's actually, when it's counting reads, when it's assigning reads to transcripts, it's assigning using a probability model, essentially. So, and that allows, essentially, what you're saying, what you're discussing, it allows to, it does allow to do a normalization that's specific to each length of transcripts where you have a gene that encodes multiple isoform. It allows to do this normalization. The bottom line is that this process is a bit smarter than just counting. Its probabilistic model goes into place. There's much more to this. I mean, it's basically trying to reconstruct, trying to reassemble transcripts from short reads, and what that means in practice is that you have to build a graph of connectivity between exons, right? You have to look for these reads that span across exons, and that allow you to reconstruct, pretty much reassemble transcripts. This is what's kind of shown here, in terms of paths through this exon junction graph. Yeah. And pretty much, what these tools are doing is to look at all the multiple paths that exist in terms of isoform, isoform sort of shapes or paths. And then once the tool has found this different path, is to align, sort of map the reads using a probabilistic model to each of these possibilities, and that's how the quantification is done. So it's like a multi-step quantification process that the tool Coupling is doing. Again, this is a kind of a moving target. Tools that do that better and better, are coming up all the time. That being said, we use Couplings quite a bit in my lab, and again, the command line is similar to what you've seen before, using multiple CPUs and takes the count file and outputs quantifications for transcripts. Yes.
So from Couplings, you can get the transcript level data. You can also class that gene level data.
Yeah, that's right.
Do you think that there's an advantage or disadvantage using Couplings for gene level data versus using counting method, or do you think they're?
Yeah. So there's been a long there's been lots of discussions about this whether how exactly to take the output of couplings and which output of couplings to use in practice. What we use in my lab is actually the FPKMs that come out of couplings, but there are also counts that come out of couplings that you can use for analysis, including DEC, some of the count-based approaches to looking for differential expression. I mean, the question is more, how do you go from couplings to differential expression? And that, there are multiple ways to do it. Couplings as tools to estimate differential expression. They are pretty much universally seen as not so great. It's just the model that's using couplings, CuffDiff, that's the tool that's used, is actually not so good. Tools like DEC, are better at modeling read count, distribution of read counts. Typically using negative binomial approaches with some additional normalization steps that help with it. But yeah, so this is still a moving target. But I do want to say that in my lab we use FPKMs, confidence, and then we do things like lima, things like t-test, differential expression, which might not be the way that other people want to do this, but this is what works for us basically. Yeah, so this is another tool here for scripture. This is also these are tools that are reasonably often used for transcript quantification, although much less in the couplings. I mean, we never use them, but they do appear to have certain issues about reliability. So at least people not using them as much as other tools. This is just to illustrate illustrate something that's going to be covered in much more detail by Naomi about all of the biases that one can find in Illumina sequencing. There are many, many biases, many, many issues, many problems that have to be taken into account. This is just reasonably recent papers that look at some of these problems. This is a paper that pointed out a problem that's coming from priming the random primers that are used for sequencing. Just creating some biases that, again, one have to take into account if you want to have very reliable quantification of expression levels. This is also an active field of research that maybe will tell you more about something. So quickly, let's discuss single nucleotide variation analysis. This is looking for mutations. It's basically just using the fact that when you align short read to the genome, to the reference genome, sometimes you have a perfect match like this, but not always. Sometimes you have mismatches. One of the issues is that sequencer still makes a lot of mistakes. The question is how to distinguish mismatches from actual differences between genomes. Again, everything is aligned to the reference genome typically, which is the consensus of 8 genomes. So you're going to find differences, due to the fact that there are differences between people, in terms of genomic content, but also the sequence is making a lot of mistakes. So you have to take that into account. When it comes to mutation detection, what you want to do essentially is to have multiple reads that give you independent information about the same position in the genome. This is just a representation here where you have each line here is a read. Each dot here corresponds to a match between the read and the reference genome. When you have a mismatch here, it shows the nucleotide that's mismatched. You can see here you have multiple reads, and all of these reads share a mismatch with the reference. This is an easy situation here where you have it's a very clear variation here. This is a difference between the reads and the reference. This is a 0. Obviously, this was a homozygous mutation, homozygous variation. 2 copies of the same chromosome have different variations. Very often, what you have is heterozygous variations, right, where you have about half of the reads that have a variation, the other half doesn't. Right? And you see all of that is in the context of a lot of sequencing mistakes, surrounding this position here. So the detection of these becomes a little bit trickier in the context of all this. Are these real biological variations or are these sequencing errors? That's definitely something that is not always easy. And the difficulty is even worse when you have low coverage, where again you have situations like this, very few reads. You have half of the reads that have a variation. You have some isolated mismatches like this. Is this a real biological variation, or is this just a set of sequencing errors that can line up in the same location? That's something that needs to be figured out. And things are even worse when you try looking for cancer mutations. Trying to detect cancer mutations from short reads. You are basically looking at tumors, where there's a lot of heterogeneity. Some of the cells are going to have mutations that are going to be shared by all of the tumor cells. This is actually an easy to detect. But if you have a small subset of cells that have a mutation, that's going to lead to a situation where you have very few reads that have a mutation. And again, detecting these as mutations as opposed to just sequencing errors is a challenge. And again, you have all kinds of things happening in cancer cells, including loss of genetic material that give rise to loss of heterozygosity. So you end up in situations like this where you have homozygosity due to this loss of genetic material. Just to illustrate the fact that especially in tumor cells, tumor sequencing, you get all kinds of situations. And those are pose challenges in terms of mutational protection. Together with the fact that you have, as we saw from the quality score plot, the quality scores, as you remember, were declining as you were moving away from the beginning of the reads. What that means in practice is that you see more and more mismatches towards the end of the reads. And therefore, if you look at mismatches as a function of the position of the reads, you do see more mismatches towards the end. So again, if you have multiple reads, the same location, that overlap a single nucleotide, but the mismatches are always towards the end of the reads, right? I mean, the question is, is this really a biological variation, or is it a mutation? There are tools to pull single nucleotide variations. GATK is one of the most common. There are also tools such as VASCAN that are used quite a bit. Other tools that are specifically used for cancer detection for mutant. We do have a mutation called MLab. I'm not going to go into detail on this. It does model this unequal rate of mismatches across the read. Not too much time to go into detail about this. This is something that I talked to you guys a little bit. The fact that it's hard to call indels using deep sequencing short reads. And the reason is, that most often these indels occur in microsatellites. Let's say that you have an insertion. Let's say that you have a read that has an insertion of 1 CA on this location, right? When you map it to the genome, you can put the insertion in multiple locations. Right? So you can put it there, and there, right? And the score of all these insertions is going to be exactly the same. Right? And what's happening is that because the way that the algorithms work is it's such that basically the insertion is going to be in different places, different reads. Even if they're aligned to the same location, the insertions are going to be in different places. So you do have to work extra hard essentially to make all of these reads agree with each other, and this is done using short read alignments around indels. This is something that GATK was doing pretty well. Just to essentially to summarize, I mean, indels index calling is a bit harder on these extra steps. These are tools for annotation. So once you find the mutations or the variations, you want to annotate them. You want to know what they do in terms of protein sequence changes. Do they impact the protein sequence in such a way that it's likely that it's going to lead to a loss of function of the protein? There are tools that can predict that with some level of accuracy, I would say. Polyphen and mutation assessor tools like this. Mutation assessor is actually a bit smarter because it looks at where in the protein sequence the mutation is happening, whether it's happening in the domain that's important for the function of the protein, whether it's hitting a highly conserved amino acid, whether it's changing the physical chemical property of amino acid, for example, going from a hydrophobic amino acid to a hydrophilic amino acid. If your mutations are such that they do change the property of the amino acid, they're likely to impact the function of the protein. Again, there are lots of tools for doing this. For doing this annotation. CellSNP is another one that people use quite a bit. Cross-species conservation is also used as a way to gauge how important a position of the genome is. And if it's highly conserved cross-species, I mean, chances are that changing that nucleotide that's going to change the amino acid is going to be deleterious as well. So this is short read visualization. So this is a tool in SAM tools that allows you to do a text-based short read visualization. It's actually very reasonably useful. If you work in the command line, and if you have your BAM file that's sorted and indexed, this allows you to very quickly look at a location, look at your reads. It's pretty useful. It's faster than having to fire up IGV or load your reads into a genome browser. It's just text-based visualization. You can see the mismatches here. You can see the orientation of the reads, different codes for different orientation, comma or dots. Again, the genome browser is it should be a good friend. It's a great tool for visualization of data. You can upload BAM files to a genome browser. I mean, maybe a better way to do this because BAM files are huge, so you don't necessarily want to upload it. 10 BAM files take forever and might actually fail in the middle. better way to do this is actually to upload your BAM file to a web page to which you have access to web server essentially. Basically, to tell the UCSC Genome Browser to access your BAM file, you basically enter the address of your BAM file into the UCSC Genome Browser. It's going to be able to access your BAM file on your server, and it's going to be able to pull only the regions that you're interested in visualizing, and it's do that on the fly. So it's a very much quicker way to visualize BAM files using the Genome Browser. Probably still a little bit slower than using IGV, but again, it has a lot of advantages in the context of all of this additional annotation information that the Genome Browser has. And this is IGV, also a very useful tool for visualization of short reads. These are all short reads, and this is a free tool that's mid-level type of road. That's very, very nice. Getting better as well. So this is something you're gonna see a lot. Read densities. This is basically counting at each position in the genome how many reads map at a given location, and it basically just get a read count. So this is like units of 1, 2, 3. So that you have 1 reads that maps here, 2 reads that maps here, and so on. So you get essentially this read density plot. Typically, you want to smooth them as a way to get kind of nicer looking plots. But this is something that you'll see a lot. I think that you'll see to one that you do something that is important is that when you visualize read densities and when you want to visualize multiple read densities for multiple experiments, you have to take into account the fact that some experiments you have more reads than others. So again, if you have 20 million reads in one experiment, 10 million reads in another experiment, your read densities are going to be higher in the second experiment. So you do need to normalize read counts by the total number of reads. Kind of an FPKM or PKM-like type of normalization if you want to be able to compare them visually. Very important step when you make these read counts. So that you want to put them on the same scale essentially so that you can compare them visually. Basically just involves doing this the M normalization in FPKM, which is dividing by the total number of reads and multiplying by the this is a way to create a format which is called a Weill format that contains this read density information that you can upload to the Genome Browser. You can have tools to do this. So basically, these are genomic intervals of 10 nucleotides here, and you have the average read count. These are normalized read counts here for the interval of 10 nucleotides. And you basically have this information for the entire genome. The formats of this is the actual definition of formats, because you need to use a few additional beta tags to tell exactly what you want to to the Genome Browser. The format for these is URLs.
Are these indexable?
Yeah, so yeah, exactly. So Weill is not indexable, but the big wig is. And in fact, big wig is actually the way to go for this file because of what it's actually just huge. Big wig is the binary version of Weill, and it's indexable, and it's fast, and it's easy to access. This is absolutely the way to go because, like you say, these files are getting huge, and you can't manage Weill format. You can look at it, but you can't do anything with it. So, yeah.
Is it better to use big wig if you try to load BAM? Is it faster to use
Yeah, so this is the problem with bigwig is that or Weill is that you collapse all your reads into read density, so you lose information about mutations, whether the read maps in the forward or reverse strand. So it's complementary. So this is giving you useful information for some aspects. Basically, it allows you to look at your reads. The raw data is coming out of sequencer. But obviously, this is also a bit messy, and because there's too much information, the Genome Browser has to pack up, essentially to pile up these reads here in ways that are not always very reliable visually. If you were to zoom out, you would actually see it. These reads actually don't, pack up, necessarily in this way. If you were to zoom in, so you would see that they are actually. It's just the way that the software works in terms of displaying those reads in the same picture. Right? It's just not visually reliable, basically. But it's useful as a way to get a sense of what your data is like. Again, visualization, is key, and visualizing what data is very important, and that allows you to do it. This is collapsing information here. By doing this collapsing, you lose information. It just tells something else. This is just an example here. This is actually showing the two things next to each other, the reads and the actual densities. And you can see, what I was telling you is that this actually doesn't quite reflect what you're seeing here. It's just kind of artifact to the way that the reads are piled up. But yeah, that's some great news. All right, these are BioConductor packages here that are very useful when it comes to deep sequencing data analysis. These are packages that tomorrow will introduce you to. iRange is a great package for operations on intervals and ranges. RSAM tools, interface to SAM tools. Short read allows you to read in a lot of different read formats into R-track layer, allows you to play with a genome browser. It's actually a very cool package that allows you to interact with a genome browser from R. It allows you to upload tracks to the genome browser and basically kind of fire up the genome browser from R, and you can add tracks. You can change the location, the window that you're looking at. It's a very cool package. The BS genome allows you to get information about different genomes, so you can see what the reference genome is available. And again, there are many more packages for deep sequencing analysis. I do want to say, and I think I said that, but I don't want to reiterate that, you can also execute command line programs within R, just using a system command. That allows you to have workflows that are entirely R-based, even though you're using external tools, and you have to load in the output files into R eventually. But not all deep sequencing packages have a native R interface, and using this system command is very, very helpful. This is a great link here. This is a link to I think that Sean, you mentioned that already, this URL. This is a page, it's almost like a book now, that lists all kinds of analysis that you can do from R on deep sequencing data. That includes, things like that we discussed, like trimming of linking sequences, adapters, and so on and so forth. You can do all these things in R. Whether it's the fastest option or not, this is always a matter of debate. But it allows you to have workflows that are entirely R-based, and that's beneficial in terms of maintaining your code and having code that can evolve, and it's just easier. It just makes your life easier. This is just quickly. Some of the issues that we're all facing when it comes to deep sequencing data. A huge increase in sequencing speed and throughput. That really is outpacing some of the other increases that we see. For example, the Moore's law. Definitely, huge problems in terms of data storage, and that we're all facing. That's the reason why, there are, also kind of ways to deal with this. That there are all of these now these formats for storing short reads. Typically they're compressed. There are actually short read formats that are specifically optimized for short read compression. now there's a successor of Salmon BAMS, which is called the CRAM format, which really does a very, very good job at compressing information from short reads. Trying to really minimize the size of his files, maintaining accessibility, maintaining this indexing scheme that allows you to quickly pull different slices of a data set into analysis of those. There's reduced BAM, which is also a format that people use more and more. And sequencing is becoming faster. This is a MySeq, I'm sure most of you have heard about, that allows you to just sequence a lane, not a full lane, a kind of a small lane, but 20 million reads or so in just a day. And it's really changing the game when it comes to sequencing genomes, sequencing clinical samples, and so on. But it also means that you're getting a lot of sequencing data all the time, and that's challenging in terms of analysis. And technologies to make those reads, to create longer reads. There's challenges in terms of clinical application of sequencing. This is assembly. We talk about assembly. There's lots of people working on trying to assemble de novo transcripts from INDEXing data. Not a simple problem, but solutions are getting better and better. Data integration is a key problem too. As I was telling you in some of the other slides, showing you that people do kind of experiments on the same cell lines, same tissues now, and major challenges in terms of integration of these multiple data sets. This is something we've done actually in my lab, integration of CheapSeq and INDEXing. Basically, we integrated statistical models of using the binding information to try to predict how genes are expressed. Essentially, that gives you, we use smart algorithms and concepts like principal component analysis, non-negative matrix factorization that allow you to really kind of capture correlations within your binding data and kind of correlate this using models to expression levels. That in turn gives you additional predictions and that leads to the potentially new experiments that you can use to improve these statistical models. This is the way that data integration works, essentially, merging things into using a statistical model. And that's it. This is I'm happy to answer any questions that you would have. Anything else? Anything that we discussed?
Maybe we should discuss over lunch.
Yeah.